Hi, my name is Harriet Kamendi, and I am the CEO of Candy Bioscience. At Candy Bioscience, we provide regulatory toxicology support to developers of MedTech products. So, in this presentation, I want to go through the things that you must be thinking of when you're developing a medical product. So, below is the anatomy of MedTech risk, from non-contact software to very high impact risk implants. And we're going to be going over regulatory frameworks, patient safety ecosystem, and the quality management system that is at ISO 10993-1. So before we start, I guess it's important to discuss why it is so imperative that we assess risk of products that we are developing. The cost of medical complexity is quite high. So every year, 3 million people are hospitalized due to different medication imposed or even device imposed issues that arise during use. 106,000 of those individuals will die. And this costs the government a total of $528 billion. 528 billion US dollars, and that's quite a systemic healthcare risk that is behavioral and systemic and not necessarily biological. So we have to manage complicated diseases using a whole slew of solutions, for example, drugs, food, dietary interactions, medical devices, and we have to figure out how all of those will interact with the complicated ecosystem that is our bodies. So when you think about devices, you have to think critically about the risk they pose. And you have to start by determining what kind of device are you bringing to market. For example, on this slide, we have a software as a medical device. So think about these Apple Watches that are now measuring blood pressure. And the risk in that case is one that is very low. However, you still have to think about the kind of information it's providing and what that information depicts to the patient and what the implications are when the patient receives that information. So in this particular app, in this particular software as a medical device, it's an app that you download. It doesn't interact with the individual. So the behavioral risk involved is almost zero because there is no physical contact. Therefore, when assessing risk, you have to begin with the contact continuum. Yeah. So is the device going to be a digital app or is it going to present physical risk? And when we are thinking about the devices, then a downloadable app is definitely going to be different from a catheter or an implant. And therefore, the way you assess risk is going to be different than in one versus the other. For example, the nature of contact for the software is non-contact, for the implant and catheter is direct or direct, sometimes indirect tissue contact. For the risk profile, the software is behavioral algorithm. For the risk profile that is physical, it's going to be a biological, a toxicological response. Therefore, when you're considering what FDA is looking at, the constraint for the app is software validation and cybersecurity, while that for the implant is a quality manual governed by ISO 13485. Then that means that for biocompatibility, it's waived for the software as a medical device, but for the catheter and the implant, there'll definitely be a rigorous ISO 10993 process, depending on the contact that the device will have. So the core question is what happens when an innovation actually touches the person or the skin? So one of the key tenets of MedTech device development is the physical baseline governed by the Quality Management System Regulation or the QMSR and ISO 13485. Now, this tenet suggests, no, this anchor says that the biocompatibility must be conducted in a fully developed device that has gone through manufacture, and therefore it suggests that if your manufacturing process is stringent, then your device will be safe, effective, and compliant. One, and two, it will be sustainably produced to help the intended people. So the governing principles or the guidances that are used to guide the development of medtech products, that's the ISO 10993 risk management protocol. One indicates that the product must be evaluated in its final form. So the product must have gone through full manufacturing all the way to the end, packaged and delivered, ready for sale. Then you assess the risk. So how do you begin the risk assessment process? Well, first, you must identify the raw material. You must figure out if the raw materials have got any liabilities that accompany them. Then you can go ahead and conduct a chemical toxicity and mechanical failure hazard assessment. And that is taking a look at what is leached or extracted from a fully formed, fully completed, fully manufactured device. Then once you review that data against existing literature in a toxicology risk assessment, you can go ahead and identify gaps that you then need to use to figure out a plan for in vitro and in vivo testing, which will form the basis of biocompatibility testing and enabling you to to generate data that can then support your registration application. So, depending on the class of device and the perceived risk, then you can leverage various types of data, including literature. So you can use the toxicology thresholds derived from literature sources, and you must be looking for information that aligns with the intended use for the product you're developing. Then you can also generate animal study data that comes together with in vitro assessments, and this can substitute specific tests, at least where you need to add more information to enable risk assessment. Clinical experience is also indicated in some instances, for example, for class 3 devices, and generally it's conducted on a few patients, so the signal is not as robust, but it gives you time. It gives you data that can help in framing your risk to the regulator and going forward. And then previously reviewed devices, you go into the FDA literature database and you can find previously approved devices and read summary statements from the FDA that can guide how you think about risk of the product that you're developing. It doesn't mean that what you get is going to be aligned with what you're looking for, but it tells you what the FDA is, how the FDA is thinking about selected devices. And it is important when you're looking through these databases to go back to figure out exactly what guidance that device was developed under, because there's a continuous change in how the FDA views issues, and they write guidances and update them regularly for such that a device from 2025 is definitely different than a device from 2015. And in that, the FDA has updated the guidance to reflect its current thinking. So FDA has been marching towards the 3R transformation, which was initiated, I believe, in 1959. and therefore they minimize the use of animals in testing and only use them where absolutely necessary. So the framework for risk assessment initiates with literature reviews, minimal testing, for example, characterizing or doing chemical characterization of extractables and leachables that are derived from the fully finished device. And that is the preferred first line of defense or first line of information. Because if you do not find any reason to continue testing, then you don't have to add additional testing. So then you have the in vitro models. There are those that are mandated, like cytotoxicity, irritation, and sensitization. Those are mandated and you have to do them. But they end there, and animal studies are kind of the last resort that is that you use to justify risk or to assess risk of your product. So as you develop your product, it's always good to imagine or to start from the very top and proceed systematically, reducing animal use as much as possible and using it only when it's needed. So other factors to consider are the nature of contact. So you have to consider when you're looking at the device that you're working with, is it going to contact intact skin? Then there is low risk. Is it going to contact a blood path, a tissue, or a bone? And if it does, then you have to worry about activating the immune system. So the testing burden increases to enable you to assess risk appropriately. And the last part is the implant or a device that is going to be implanted into the body and last a while. That you have to consider testing that has to demonstrate that that device in the body is not going to, if anything, cause cancer. So the amount of testing, the amount of testing or the burden of testing increases based on how the device will interact with the body. The other factor to consider is the time limit in which the device will be on the person. So if the device will be there for less than 24 hours, low burden. If you're going to be there for a limited time, subchronic exposure, 24 to 30 days, then again, short duration of testing. But if you're going to be around long enough, then the burden of testing increases, especially if, again, if you're going to have the implant for a long time, then we're talking full-blown risk characterization with all manner of testing to support the device's risk assessment. So for a device, say for example, a knee replacement or a hip replacement or a stent, for the cardiovascular indwelling stent, you have to assess whether it leaches anything, how much every day, and then you then calculate the risk of how much it's going to expose the person to over a lifetime. If that implant is going to be in the body for years, then you have to assess risk for that duration of time. And then, of course, when you're testing in animals, you have to consider that animals have a shorter lifespan. So you really don't need to do 30 years on a rat. A year on a rat gives you enough information to determine how the implant is going to behave in a human 10 years from today. So when you're thinking about your device, you have to think about what the FDA is actually after. So they want the final finished form. Why is this necessary? So if you're building, say, a hip joint, yeah, it is formed, say, from a titanium, which is a metal, but that titanium is going to be heated, machined, sterilized, and all of those activities can change the surface of the device. It can, for example, some things can be left on the titanium, or over time, it can probably leach something into the blood, and we need to determine what those leachables are. And therefore, when we are taking the final finished form, it has gone through all the processing that it needs to go through, and everything that a patient is likely to be exposed to exists on the final form. And so you can take that and you can actually assess the risk of the final product in the manner in which it will be implanted into the patient. If it's an implant, or the way it will be, it will get in contact with the patient when it is in its final use. So if the product is not in final form, there is no use of testing. It doesn't really make sense, or it doesn't give enough information. So how do we conduct chemical characterization? The first thing is we have to leach or remove the leachables from the device. So if you think about the body is composed of water, but it's also composed of fats. And therefore, when you put the device in the person, there will be an interaction between the device and water, and a device and fat. And so you have to literally extract the device in both a polar solvent, which represents water, and a non-polar solvent, which represents fat. And what the components that leach out of this are what you're going to be assessing. The other factor to consider is that it could be because you're only going to test it for a brief moment, you want to exaggerate the conditions. So, for example, common protocols allow us to heat the product up to 50 degrees for up to 72 hours to accelerate the extracts and the leachables that might come out of the device. If that is not possible, or if it is expected to over extract, then we can fall back to 37 degrees for the same number of days. So this is normally done for products that are not heat stable. For example, if you have a collagen product and it's likely to degrade completely in 50 degrees centigrade, then we can justify using 37 degrees for this component. And then once we extract, then we can use our LCMS, GCMS, and ICP to identify the components that are present in our leached solutions. So two assays that we normally do. Let's take a moment and look at them: sensitization and genotoxicity. There are two kinds of assays in both. You can either do the in vitro or you can do the in vivo. And with the sensitization assay, let's do a little bit of a deep dive. Sensitization assay is mandatory. It has to be conducted for most characterizations. So there are two: the local lymph node assay, which is in vitro, or the guinea pig maximization test, which is the in vivo. So the LLNA is used for aqueous solutions as well as for metal compounds, while the GPMT, or the guinea pig maximization test, is used for novel materials and those materials that will be contacting deep tissues, or those that are known to contact blood vessels or to trigger an immune response. This is a mandatory test. There is no way around this one. The genotoxicity assay, however, is not mandatory, and it's mandated when your data shows that you might have a signal. And so there are the two most commonly used are the in vitro assays, which include the Ames assay, or the bacterial gene mutation assay, and a mammalian genotoxicity assay. And both, if you pass both, you don't have to go any further. If you don't, then you have, if the one of them is inconclusive, then you have to do an in vivo assay as a follow-up. The good thing about the genotoxicity assay, you only do when you identify a signal following chemical characterization, and the toxicology risk assessment flags that there's a problem and that there is likely to be a genotox signal. If there's a genotox signal in your chemical characterization and toxicology risk assessment, then the genotoxicity assay is mandatory. Another set of assays that is required for pretty much all medical device class categories or other biocompatibility testing is a cytotoxicity and irritation assay. Cytotoxicity evaluates the potential of materials to cause cell damage or death. There are multiple assays, and each assay is selected based on the material type, the extraction method, and the clinical contact duration. Irritation studies are done on the skin or the mucosal system, depending on whether the device contacts the skin or the mucosal surface. And there are assays for each type of contact, and the scoring is conducted based on a published guidance. And again, it aligns with the contact, the material, the contact duration, the extraction, and the clinical use. Acute and chronic toxicology studies are a different category of studies that are conducted when there is a risk identified, short-term or long-term risk identified for the device you're testing. Acute studies are single doses. Subchronic are repeated doses over a period of 28 days, 28 to 90 days. And then chronic toxicity studies are those that are required for products that will be long-term in duration. Another category of studies, before I move on to that, acute and chronic studies can be waived following the conduct of a chemical characterization as well as a toxicology risk assessment that determines if there is a threshold of toxicologic concern. Implantation studies are conducted to evaluate a biomaterial or device in the body that demonstrates that will be in the body for an intended duration of use. They are conducted per guidance, and they will be implanted into the skin or the muscle and characterized fully for evaluated over a duration of days. So what about those products that are contacting the vascular environment? Those ones present systemic risk. There are two kinds of assays. One, hemocompatibility, and that is there are two kinds of tests that would be in this situation: the direct contact and the indirect contact. When you have a product that is going to be directly interacting with the blood vessel, then you would have to do two tests: the hemolysis and the thrombogenicity test. While those ones that are indirectly contacting, like wound care products, where there is a wound that is broken, there are blood vessels visible but not directly contacting, then or that the product can leach into a blood vessel, then in this case we need to do indirect contact testing, and that requires us to do a hemolysis test only. Devices subject to high shear stress, say for example those ones that are going to sit somewhere near the aorta, then they require dynamic in vitro assessment under simulated clinical flow conditions. So you do the direct assays, the hemolysis and the thrombogenicity, but in a situation that reflects clinical or human blood flow, and in order to satisfy that requirement. The second kind of testing that must be done is pyrogenicity, and this is fever. So you're protecting against febrile reactions. These are normally caused by bacteria. So you are testing for endotoxins in the final product. And that's done as part of the sterility testing or right after sterility testing. I mean, sterility, the sterilization has happened. So you will go ahead and assess for endotoxin in the final product. Material mediated is chemicals that leached from the device. Is there any that would cause febrile responses in a rabbit? This test is conducted in rabbits only, and is part of the protocol required for devices that are contacting the vascular environment. So for devices that are complex, for example, nanotech or degradables, then there are two factors that we must look at. One is that for that one that will be in situ, polymerizing and absorbable, and which is absorbable in nature, for example, collagen, or for example, these gels, that hydrogels that are used that behave like materials, or that are regulated as devices. That's it. So in situ, those materials must be evaluated at time zero, and then you pick a time in between the final test and assess changes over time. And then at the end, and the reason you do this is you want to determine if the material breaks down during the study, and also assess whether there is any risk that comes up, and then you assess at the end again to do the same. So biocompatibility in vivo is tested at the intermediate and final time points. The other thing is that in the submicron and nanotechnology, for those ones that are less than 1 micron, you need to do chemical leachable to see if there are any artifacts of the device itself. And then there are unique concerns of absorption and distribution, and therefore you need to see if these particles will cause any kind of irritation and or encapsulation near target organs. So then the medical technology risk and innovation spectrum spans a whole host of a wide variety of situations. So in the first place, we have software, and that only poses a behavioral risk. It's virtual, and there's zero biological contact. The only risk we are thinking about is the code validation and UI design, and it solves a massive systemic cost. So the surface contacting devices rely on a QMSR, and that's the quality management system. And these include wearables and external biosensors. They contact the intact skin, so there is very low biological risk, and they rely on ISO 13485 manufacturing consistency, and most of this is in vitro. They rely on in vitro chemical characterization. The long implants, biological risk is high. These are stents, absorbables. The maximum biological risk is significant. These require full ISO 10993 suite, genotoxicity, hemocompatibility, and in vivo testing. And so when you're developing these types of products, then the patient safety requires mastering, and there's a continuum of engineering and toxicology that enables you that, and you have to be thinking of how to prevent behavioral errors and execute on a flawless physical chemistry before contacting the regulator. Thank you. So please leave a comment below or contact us at the following. Thank you.